In this episode, we enter Genome City, where the story of bacterial genetics unfolds from the very core, the circular double-stranded chromosome. Inside chromosome HQ, the bacterial chromosome operates as the master blueprint, tightly coiled yet highly organized within the nucleoid region. Unlike eukaryotic chromosomes, bacterial chromosome is typically circular and replicates through a semi-conservative mechanism, ensuring that each daughter cell inherits one parental strand and one newly synthesized strand. Complementary base pairing, A with T and G with C, safeguards fidelity during replication, while coordinated enzymatic machinery ensures rapid duplication, allowing bacteria to multiply efficiently. From this central genetic archive arise the instructions for metabolism, structural components, protein synthesis, virulence factors, and even antibiotic resistance determinants. However, the chromosome does not act alone. Plasmids are extrachromosomal autonomously replicating circular DNA molecules that provide accessory advantages rather than essential survival genes. These plasmids dramatically enhance bacterial adaptability. Our plasmids encode antibiotic resistance genes such as beta-lactamases or efflux pumps. Virulence plasmids carry toxins and adherence factors that enhance pathogenicity. Conjugative plasmids harbor the machinery required for horizontal transfer via pilus formation. In contrast with non-conjugative plasmids, TRA genes, short for transfer genes, are located on conjugative plasmids. These genes encode the machinery required to initiate and complete DNA transfer between bacterial cells. A bacterium carrying a conjugative plasmid expresses the TRA operon, which directs the formation of a specialized structure known as the sex pilus. This pilus extends outward from the donor cell, functioning like a grappling hook that attaches to a recipient bacterium lacking the plasmid. Once attachment is established, the pilus retracts, bringing the two cells into close contact and forming a conjugation bridge through which DNA can pass. A single strand is transferred into the recipient cell while replication occurs simultaneously in the donor. As the single strand enters the recipient, complementary strand synthesis restores the plasmid to its double-stranded circular form. By the end of the process, both donor and recipient possess a complete copy of the plasmid, effectively converting the recipient into a potential future donor. Clinically, conjugation plays a major role in the rapid dissemination of antibiotic resistance genes among bacterial populations, rapidly transforming a benign bacterium into a multi-drug resistant pathogen. Adding another layer of mobility are transposons, or jumping genes, capable of moving between chromosomal and plasmid DNA. These mobile genetic elements often carry antibiotic resistance genes and can integrate into multiple genomic locations, promoting the accumulation and dissemination of multi-drug resistance traits. Their movement does not require extensive sequence homology, making them especially potent drivers of genetic rearrangement. Let's shift to bacteriophages, or simply phages, are viruses that specifically infect bacteria, and they are among the most abundant biological entities on Earth. They act as microscopic predators that regulate bacterial populations in nature and shape microbial evolution. Structurally, a typical bacteriophage consists of a protein capsid head containing nucleic acid, either DNA or RNA, a tail structure, and specialized tail fibers that recognize and bind to specific receptors on the bacterial surface. This specificity determines the host range of the phage and explains why certain phages infect only particular bacterial species or even specific strains. The infection process begins with attachment, where the phage tail fibers recognize and bind to bacterial surface receptors. This interaction is highly specific, functioning much like a lock and key mechanism. Following attachment, the phage injects its genetic material into the bacterial cytoplasm while the capsid remains outside. At this stage, the viral genome takes control of the host's biosynthetic machinery, redirecting it toward viral replication and protein synthesis. Once inside, the phage may follow one of two major pathways, the lytic cycle or the lysogenic cycle. In the lytic cycle, the phage genome is actively replicated, viral proteins are synthesized, and new phage particles are assembled within the bacterium. Eventually, phage-encoded enzymes degrade the bacterial cell wall, leading to cell lysis and release of numerous progeny phages. This explosive process not only destroys the host bacterium but also amplifies viral spread to neighboring cells. In contrast, during the lysogenic cycle, the phage genome integrates into the bacterial chromosome as a prophage. In this dormant state, the viral DNA replicates passively alongside the host genome without immediately harming the cell. The bacterium continues to grow and divide, silently carrying the prophage within its DNA. In general, bacterial variation is classified according whether changes are phenotypic or genotypic. Phenotypic variation reflects environmental adaptation, which includes reversible alterations in gene expression without changes in the DNA sequence. For example, bacteria may alter surface protein expression or metabolic pathways in response to nutrient availability. In contrast, genotypic variation results from permanent alterations in the DNA sequence itself. These mutations are heritable and irreversible, forming the foundation of long-term evolutionary change. Only genotypic changes are passed to progeny, while phenotypic responses disappear once environmental pressures are removed. Within the mutation laboratory, we explore the mechanisms generating genetic diversity. Mutations may arise spontaneously during replication or may be induced by mutagens, including ultraviolet and gamma radiation, as well as chemical agents such as alkylating compounds. Point mutations involve the substitution of a single nucleotide, potentially producing silent missense or nonsense effects. Insertions and deletions can cause frameshift mutations, which can profoundly alter downstream protein sequences. While many mutations are neutral or deleterious, some confer selective advantages, particularly under antibiotic pressure, thereby driving natural selection in clinical settings. Horizontal gene transfer further reshapes bacterial genomes. Conjugation occurs when bacteria possessing conjugative plasmids express TRA genes that form a sex pilus, establishing direct cell-to-cell contact and enabling DNA transfer. Transformation allows competent bacteria to uptake naked DNA fragments from their environment and integrate them into their genome through recombination. Transduction involves bacteriophages inadvertently packaging host DNA and transferring it to new bacterial cells. Together, these mechanisms allow genetic material to spread rapidly across populations, even between different species. We enter the recombinant DNA technology lab, where science moves from observation to innovation. Inside this high-tech laboratory, genetic engineering begins with precision tools known as restriction enzymes and DNA ligase. Restriction enzymes are molecular scissors that recognize specific DNA sequences and cut them at defined sites. A circular plasmid vector is carefully opened, creating compatible ends ready to receive a foreign gene. At the same time, the desired human gene, such as the insulin gene, is isolated using the same restriction enzyme to ensure matching ends. This strategic cutting creates complementary overhangs, allowing the foreign gene to align perfectly with the plasmid backbone. Once the desired gene fragment is positioned inside the opened plasmid, another essential enzyme takes the stage, DNA ligase. Acting like a molecular glue, DNA ligase seals the sugar phosphate backbone, forming stable phosphodiester bonds that permanently join the inserted gene to the plasmid vector. The result is a recombinant newly engineered plasmid carrying genetic information from two different sources. This recombinant plasmid can then be introduced into a bacterial host cell through transformation. Inside the bacterium, the plasmid replicates autonomously and expresses the inserted gene, effectively turning the bacterial cell into a microscopic factory capable of producing a specific human protein. Our story now transitions to the applications hall, where the true impact of recombinant DNA technology is revealed. One of the earliest and most transformative achievements of this recombinant technology is the production of human insulin. Before recombinant technology, insulin was extracted from animal sources, sometimes causing immune reactions. Today, genetically engineered bacteria produce pure human insulin, revolutionizing diabetes treatment and dramatically improving patient safety and accessibility. Another powerful application is the recombinant hepatitis B vaccine. Instead of using whole viruses, scientists insert the gene encoding the viral surface antigen into yeast or bacterial cells. These cells produce viral proteins that stimulate immunity without causing infection. This approach not only enhances safety but also demonstrates how genetic engineering can prevent disease by teaching the immune system to recognize pathogens before exposure. Finally, the horizon expands toward gene therapy, the therapeutic correction of genetic disorders by introducing functional genes into patient cells. Using viral or non-viral vectors, defective genes can potentially be replaced or supplemented, addressing the root cause of inherited diseases rather than merely managing symptoms. Though still evolving, gene therapy represents the frontier of molecular medicine, where DNA itself becomes the therapeutic agent. In Prokaryote Genome City, DNA is not merely a static code of life, It is a tool that can be edited, transferred, and harnessed for human benefit. From cutting and ligating genes in the laboratory to producing life-saving medicines and vaccines, recombinant DNA technology transforms bacteria from simple microorganisms into powerful biotechnological allies. In this city of genes and enzymes, innovation is written in nucleotides, and DNA truly becomes destiny.